Okay. No, it's all good.
Oh.
Okay, so here's how this is going to work. Morgan's going to be covering the first topic. I'm going to cover the middle three topics, and then Morgan's going to get the last topic. Whoever's not speaking is going to be fielding questions in chat. So if you guys have a question, feel free to ask in real time, and someone will type a response to you. If anybody here, like physically here, has questions, just raise your hand. We'll get to you. Yeah, this is being recorded right now on Zoom, and the slides themselves will be sent out by email, right? Yeah, we'll send you guys the slides. And if you have any questions, yeah, this is supposed to end around 9:30, so we're gonna try and leave time at the end for questions, and fingers crossed, all that. Yeah.
So topic 1 was intro to cells. Hopefully you guys are pretty good at this topic by now. We're just going to run through the basics again. This is mostly going to be content review with a few questions thrown in there. So starting off with eukaryotes and prokaryotes, we want to know the similarities and differences between these. So this should all be reviewed, but eukaryotes they have membrane-bound organelles. They're usually multicellular, but not always. They have linear DNA enclosed in a nucleus with a membrane, and transcription and translation occur separately. And then in prokaryotes, they lack membrane-bound organelles. They're unicellular. Each cell is its own organism, like bacteria. They have a circular DNA free-floating in the cytoplasm, and they may contain plasmids, which are those small circular pieces of DNA. And as we know, transcription and translation occur simultaneously here because, again, there is no nucleus. And then the similarities would be that they contain genetic material, which would be DNA and RNA, and then some prokaryotes will have DNA or RNA. And then they each use transcription and translation, and they each perform their own metabolic processes that we learned about in class. And then they can undergo cell division, and some have flagella, which is that little tail that moves them along. And not all do, but they both can have that. So gene expression. Gene expression is the process where the information contained in genes begins to have an effect and is physically available in the cell. Genetic information must be written in RNA, and then through transcription and translation, we see it expressed as proteins. So again, DNA to RNA to proteins. And eukaryotes, this transcription occurs in the nucleus, and then translation occurs in the cytosol, where the ribosomes are located. This means that the mRNA transcript produced and transcription has to move from the nucleus to the cytosol. And again, this doesn't have to happen in prokaryotes because they don't have a nucleus. So any questions so far? Okay. And this is just a little bit on transcription. Joe's going to go more into that on the whole unit on transcription, but this is DNA to RNA again. Again, this happens in the nucleus in eukaryotes, cytoplasm in prokaryotes, and you start with a double strand of DNA, as you can see in the image right here. And then RNA polymerase reads the non-coding strand, which we know is the template strand, and creates an RNA transcript. So the first question of the night for you guys is, which of the following is true regarding transcription? Just read these, and if you have an answer, raise your hand.
You guys can also answer in the chat if you'd like.
Yeah. Any thoughts? Yeah. Yes, that is correct. So do you want to say why it's correct? Okay, yeah.
It's fair. Yeah.
Yeah, so this one, you can kind of remember this from the lac operon unit, where she briefly mentioned that RNA polymerase does not transcribe all 3 genes at once. It actually dissociates in between and then comes back down to transcribe each individual gene. Just a good way to remember that genes are transcribed separately. And yeah, the other one, elimination, should be elongation. Transcription doesn't use both strands, and then RNA polymerases control transcription, not DNA polymerases. Okay, so a little introduction to translation. This is RNA to proteins, RNA to amino acids and proteins. So the location is in the cytoplasm for both prokaryotes and eukaryotes. And mRNA docks in the ribosome. So the ribosome is made of protein and rRNA, ribosomal RNA. And rRNA catalyzes this peptide bond formation, which we will get more into. And tRNAs carry these amino acids into the 3 spots of the ribosome, which are the A, P, and E sites in that order. And they have portions that base pair with mRNA, and they carry the specific amino acid that matches with the mRNA codon. And when that tRNA is attached, the amino acid is charged. So, any questions? Stem cell potency, undifferentiated cells that divide to create more stem cells and differentiates to more specialized cells. These we talk about when we're talking about the blastula and development. So totipotent means it's the most unspecialized possible stem cell. It's able to turn into extra embryonic tissue or embryonic tissue, which would be like the outside of blastula and then the placenta and all of that. It can do anything. And then pluripotent can turn into embryonic tissue. And multipotent is the most specialized stem cells. And these are what we use to make those iPS cells, and they can turn into any cell in a specific system, but not cells from other systems. You can think of these as like cells that make bones or blood. And development in stem cells, again, this is really important to remember. The Marola is the totipotent stem cells. It's right in that diagram right there. And then you can see it goes into the blastula. And in the blastula, the inner part is pluripotent and the outer part is totipotent. And then in the adult body, multipotent stem cells are the only thing you're going to find. You won't find toti or pluripotent in the adult human body. You guys want time to write something down?
The last slide.
Yeah. And again, these will be sent out after if you want to really copy and paste things into your notes or take another look. So stem cell treatments. We have embryonic stem cells, which use the pluripotent stem cells from that inner cell mass of the blastula, the embryo. This destroys the embryo in order to use them, but it is the most effective stem cell treatment. And then the other one we've talked about are iPS cells, or the endorsed pluripotent stem cells, which use differentiated cells and they are reprogrammed back to the embryonic stem cell-like state. So they're kind of put back to pluripotent. And it's not the same as true ESC cells because it's difficult to induce this differentiation and it can lead to more cancer. They just might not work as well, and they're less controversial, but they just don't work as well. But the body is less likely to reject them because again, you're taking it from your own body and not an embryo that's not yours.
Okay, so hello everybody. I'm going to be taking us through the next 3 topics, so please bear with me. Yeah, okay, ready to go? Wonderful. All right, topic 2 is amino acid chemistry and protein folding. Okay, so quickly before we get into everything, let's break down some important terms and what they mean. An amino acid is just the building block of the protein. It's what proteins are made out of. They are formed by peptide bonds, which are a covalent bond that we're going to discuss a bit later. On the right of the screen, you can see a nice structure of an amino acid. You'll notice that it contains an N terminus or an amino group on the left-hand side of the paper, followed by a central carbon atom referred to as the central carbon, an R group, which is variable. It's also referred to as the side chain. And then you will see a carboxyl group on the rightmost side that is also referred to as the C terminus. It contains a carboxylic acid functional group. Now that we have our amino acid, we know that they are bonded together by peptide bonds, which are a rather strong covalent type of bond. A polypeptide is a longer string of amino acids versus a peptide, which is just a short string of amino acids. And a protein itself can contain 1 or more polypeptides. On the right of the screen, we have an example of one right there. Okay, so as I was saying, peptide bonds are a strong covalent bond. And then this formation is catalyzed by ribosomal RNA in the ribosome when the proteins are being synthesized in translation. So you can see how a peptide bond is formed on the left-hand side of the screen between two amino acids. So you will see that the carboxylate group is forming a bond with the amino group or the amine group of the second amino acid. That's going to result in them connecting together. The C terminus of one is going to connect to the N terminus of another, and that's how you create a peptide bond. And now you have two peptides. It's also worth noting that water is formed as a product, and this makes the reaction a dehydration reaction or condensation reaction. What that means is that water is produced. And so, as I was mentioning before, amino acids are numbered from N terminus to C terminus. So if you ever see a problem of like a peptide or a polypeptide, and you're asked how many amino acids are there in this structure, you start with the first N terminus, you go then to the central carbon, and then to the C terminus, which will then be bonded to the N terminus of the next amino acid. It's also worth noting here that in this diagram, the peptide bonds themselves are highlighted in blue, so you can see. You might get asked how many peptide bonds there are in something. That would just be the number of bonds between the C terminus of one amino acid and the N terminus of another amino acid. You can also see all the different R groups. You can there's many different kinds, and they vary in what's called polarity, which refers to the electronegativity change between atoms and how hydrophobic and or hydrophilic an atom is, but we'll get into that much later. Okay, so now let's quickly go over bonds and relation to cell chemistry. These are challenging. You may have learned them before, you may not have, no matter what. I think I've learned them 3 times and I still don't always get them all right. They're challenging. So the way that I like to break it down is versus first nonpolar covalent bonds. That's just where bonding electrons are shared equally between the two atoms. There are no charges. Charges are indicative of something that is polar. So you have no charges on your nonpolar covalent bonded atoms. The electrons are shared equally versus in a polar covalent bond, bonding electrons are not shared equally. And that is due to a large electronegativity difference between atoms. So as you can see, we have chlorine here and hydrogen. Chlorine is far more electronegative than hydrogen is. So chlorine is going to have more electrons. You can also see, you also see partial charges here. And then lastly, we have ionic bonds, which is the complete transfer of one or more valence electrons and full charges resulting on ions. That's when you have a metal and a non-metal. And then polarity determines how molecules are going to interact with each other. So for example, H2O, water, is a very polar molecule. Therefore, it is hydrophilic. It wants to associate with other polar hydrophilic molecules. So keep that in mind because the cell, of course, is an aqueous environment. That means that it is water-based versus non-polar molecules. So when bonding electrons are being shared equally, you have a non-polar molecule. That molecule is hydrophobic or water-fearing. They don't like water. They don't like the aqueous environment of the cell. They want to clump or aggregate with other non-polar molecules. And as you might expect, nonpolar and polar molecules repel, but nonpolar molecules will cluster. This is known as the hydrophobic effect, which has a lot to do with things like Gibbs free energy. We don't need to get that far into it, but the basic idea is that nonpolar molecules tend to clump together to avoid having to interact with anything that is hydrophilic or aqueous. And then there's just partial charges from polar bonds interact. These are called dipole-dipole forces. And then there's transient dipoles, which are just another way of saying like a weaker dipole formed between any molecules that interact. These are known as London dispersion forces, and they are the weakest form of intermolecular forces. Yes. Good question. Okay, so a dipole refers to, or a dipole moment, I believe, refers to the interaction that occurs because of an electronegativity difference between atoms. So for example, chlorine, this right here, oh, you guys can't see. I'm referring to the one first on the left, the Cl2. You have 2 chlorine atoms. Those are going to form a weak dipole interaction because they're the same. There isn't an electronegativity difference between chlorine because you have Cl2 right there. That's 2 chlorine atoms. Therefore, they're going to form a weak dipole or a weaker electronegativity versus with the polar covalent bond, you have a more stronger dipole moment being created because chlorine is much more electronegative than hydrogen is. That makes sense? All right. Any other questions?
Which one of those are the most like stable and unstable? Can you talk about that?
So in terms of, yeah, okay. So in terms of strength, covalent bonds are the strongest. These are going to be like your peptide bonds, your phosphate bonds, things like that. Everything else that is yeah, that occur between cysteines. Your non-covalent bonds, so things like ionic bonding, hydrogen, dipole, London dispersion forces, those are all weaker than covalent bonds because they're non-covalent. So when in doubt, covalent is always stronger. If you want to look specifically at non-covalent bond strength, we can look at that as well. I'm not sure if you're going to need to know that, but we can look at it.
That's kind of like when we're talking about protein denaturation and the covalent bonds in the backbone are not breaking, but what's breaking is that tertiary structure and the other bonds that are being formed that are not covalent. Good way to remember.
Okay, right here, you're going to see the chart of the 20 amino acids categorized by whether they are polar, non-polar, contain a positive charge, better known as basic, or contain a negative charge, better known as acidic. A quick trick to remember these is that the acids have acid in the name. They don't need to memorize any. You guys don't need to memorize any amino acids. You will be given this chart on the exam. And so chemistry determines the interactions between which R groups. And to be clear, the R group determines whether a amino acid is polar, non-polar, basic, or acidic. So just like, for example, you'll notice like valine, for example, that's got a carbon attached to two methyl groups. That's very non-polar, very hydrophobic because hydrocarbons themselves are very non-polar. There isn't a significant difference in electronegativity there versus something like serine where there's an OH group, that's a hydrogen bond. There is a significant electronegativity difference there. That's just kind of how this works. And within these classifications, some amino acids can be either more nonpolar than other nonpolar amino acids or more polar than other polar amino acids. This exists on a spectrum. And just, something to keep in mind. Yes. Yeah, absolutely. Yeah, so it's less about how acidity determines polarity. It's more so that you have for the ones with a minus charge, these are referred to as acidic amino acids, just because they have a minus charge. I wouldn't be too concerned about that for this class. It's not really something you need to worry about versus the basic ones that have a positive charge. You will get this chart, and I don't think you're going to need to have to talk specifically about like the intricacies of acidic and basic amino acid properties. I would focus more so on the polar versus non-polar, which is determined by the electronegativity differences and the types of molecules present in the R group. No problem. Okay. Yeah. Protein modeling. This is pretty straightforward. I'm not sure how much you guys are going to need, but there's a backbone model. It shows the backbone. You can't see the R groups. There's a wireframe and a ball and stick model. You can see the R group interactions, but you can't see secondary structures. So your alpha helices and beta sheets, which we're going to get to, you won't see that. Ribbon model is great because it shows the secondary structure and the backbone, but you can't see the R groups. And then finally, you have a space filling model, which shows the surface contour, but you can't see the backbone path or the secondary structure. Yeah, I wouldn't worry too much about these. All right, we already went through this amino acid structure, amino group, central carbon with a side chain or an R group and a carboxyl group, N terminus on the left, C terminus on the right. So just really quick to go over what bonds hold amino acids together. We'll go one by one here. What bonds hold them together? You can type in the chat. Anyone here is welcome to answer. Yeah. Peptide bonds. Yes, peptide bonds, covalent bonds that hold amino acids together. Okay. All right, my bad. Covalent or non-covalent, everybody? Shout it out. Covalent. Yeah, I just said it. Sorry, I spoiled it. It is covalent. And what byproduct is released as the bond forms? Water. Yes, because this is a condensation reaction. Okay, so Yeah, there's a lot here. Okay. Chemistry of primary and secondary structure. So primary structure is just the sequence of amino acids held together by covalent bonds called peptide bonds. As we mentioned, water is a byproduct of that. Primary structure is just the amino acid chain with the R groups. It's just just a link of amino acids. There can be however many as you want. Secondary structure is when things get a little more complicated. You have the formation of alpha helices and beta sheets, and this is caused by hydrogen bonding between amino groups and carboxylic acid groups. These hydrogen bonds are non-covalent bonds, so these are weaker than covalent bonds. Remember, we discussed this. And lastly, important to know, secondary structure does not involve the R groups at all. It only involves the amino and carboxylate groups, which make up the backbone.
Yeah.
Yes. Sorry. Sure. Yeah, absolutely. So a hydrogen bond can only occur if you have a hydrogen bonded to an oxygen, hydrogen bonded to a nitrogen, or hydrogen bonded to a fluorine, which you will see less often, but it is true. Okay, tertiary and quaternary structure really quick. Tertiary structure is just a single polypeptide chain, and the way it specifically folds due to non-covalent interactions. Quaternary structure is just multiple polypeptide chains folded together where each polypeptide is a subunit. These are stabilized by covalent and non-covalent R group interactions along the polypeptides. So things like hydrogen bonds, electrostatic interactions, and van der Waals forces.
Yeah. And those like 4 boxes right there, those can be some like helpful bullet points you might want to put in your exam sheet if you haven't already. One for each.
Yeah, that's a great picture describing each part of structure, because each part, primary, secondary, tertiary, quaternary, has its own function, involves its own things, and are all equally important. Here's a quick bond summary slide. I'm not going to go through all of them because we kind of talked about that already, but I think this is something that's very useful to have for your exam. So if you guys want to take a picture, the slides will be sent out. And protein damage. I think this might be the last thing in topic 2. So protein folding can be highly influenced by the environment. Things like heat, light, salts, pH. What was the example in class? Like detergent? Detergent. Yeah, detergent can disrupt proteins. These all can disrupt the non-covalent bonds in a protein. And there's a lot of those in a protein. It's not just covalent bonds. So that's a problem. Things like thermal energy, too much heat means more atomic excitement. Diffusion rates go up, which isn't always good. Electrons and bonds move too much and weaken, especially in the presence of thermal energy that can cause the proteins to unravel. That's very bad. And then light, high energy photons bring in a lot of energy into bonds that can break bonds, as we've learned in the second half of the class. This can also sometimes affect covalent bonds too. That's also how DNA breaks, which is, as you might guess, very bad. And then salts and pH, these are ions. They have positive and negative charged particles that can get in and disrupt bonds, as you might imagine, based on how charge interactions work. Okay, that's it for topic 2. Great job, everybody. Any questions before we get moving on topic 3? Yeah.
Well, like, affect all the other ones.
That is a great question. It depends on where you make the change. So it depends on where you make the change, and it depends on what the change is. Like, for example, if you change primary structure, so like you're changing one of your, that involves changing one of your amino acids. So if you switch a polar amino acid with a non-polar one, big change. That's how some neurodegenerative diseases happen. But if you substitute a polar amino acid for another polar amino acid, you might not see much change. That's a good thing to keep in mind. All right, topic 3, DNA and RNA structure, transcription, RNA processing. Okay, transcription. Morgan talked about this briefly, so I'm not going to spend too much time on the basics, but basically it's just the process of converting DNA into a complementary RNA strand or polymer that can then go on and become a protein. DNA codes for proteins, RNAs can then be translated into functioning 3-dimensional proteins. RNA polymerase always reads the DNA template strand from the 3 to 5 direction, and then new DNA is always synthesized excuse me, no, new mRNA is always synthesized in the 5' to 3' direction. This is super important. You need to make sure you understand that RNA polymerase reads 3 to 5, but that new mRNA is made 5 to 3. For a gene, only the template strand is used. The non-template strand is not used by RNA polymerase. Okay, DNA nucleotides versus RNA nucleotides. There aren't that many differences. However, the biggest one is that in RNA, uracil, which is classed by U, replaces thymine, which is classed as T. In base pairing in DNA, you have GC bonds, so guanine bonding to cytosine. These create 3 hydrogen bonds when they bond together versus adenine and thymine. These create 2 hydrogen bonds. So keep in mind, it is easier to break adenine and thymine bonds than it is to break GC bonds. However, in RNA, G still bonds to C, 3 hydrogen bonds. Adenine bonds to uracil, that's 2 hydrogen bonds. And what base pairing does is that ensures that the 2 strands are complementary. So if you have a guanine base paired to an adenine, that's a problem. That is not right. The cell will go ahead and fix that through different pathways that you don't need to worry about. But that's how base pairing works. Okay, double-stranded DNA directionality. Yeah, this stuff is tricky. So DNA has a 5' end and it has a 3' end. Each strand, so you have a template strand and a non-template or coding strand. Each of them have a 5' end and a 3' end. The strands run anti-parallel to each other, and that's how they base pair in opposite directionality. That's how base pairing complementality works. DNA prefers double-stranded form. RNA prefers to be single-stranded. That doesn't mean you'll never see double-stranded RNA, but just know that it prefers to be single-stranded. And then RNA can base pair with RNA and DNA, but not as stable as DNA base pairing with itself. We going too fast for anybody? Yes. Okay. Good to know. That's fine. That's totally fine to say, guys. This is for you. All right. So do we have any questions on double-stranded DNA directionality and how it works? Good to go? All right, let's do it. Okay, transcription process. It gets broken down into 3 simple steps. So the first of which is initiation, which is where with the help of transcription factors, RNA polymerase is able to bind the promoter region in DNA. This occurs in both eukaryotes and prokaryotes. There are different types of transcription factors, which we will get into later, but the process is fairly similar across the board with a couple of nuances. The next step is elongation, which is where using base pairing between DNA and RNA polymerase is able to read the template strand and form the complementary mRNA strand that is complementary to the template. And then lastly is termination, where RNA polymerase reaches the termination sequence that tells it to stop transcribing. And that's pretty much the depth to which you need to understand transcription. Okay, nucleotide structure. So there are always 3 components to a nucleotide. Nucleotides are made up of nitrogenous bases, a sugar, and a phosphate group. You can see that in either one of these pictures. Bonds between sugars and phosphate groups are called phosphodiester bonds. These are also bonds in the DNA backbone that hold each like base pair together, not when bases are interacting. So when you have like a GC bond, that's hydrogen bonding. But when you have the DNA backbone itself, these are phosphodiester bonds. And so, although it's not shown in this image, there's an OH group attached to the 3' carbon in both RNA and DNA. So that would be just on the 3' end. You'll notice that in DNA, there is a hydrogen where on the 2' carbon, whereas opposed to RNA, there is a there's an OH group. That is the key difference between DNA nucleotides and RNA nucleotides. Those are important. Make sure you know that.
just like count in this line.
Okay, so now we're going to talk about the mechanism of transcription and how nucleotides are added. So this is really important. I said that mRNA is synthesized in the 5' to 3' end. Nucleotides are always added to the 3' end. Always, always, always. They will never be added to the 5' end, or at least not in a way that I have seen. So always they get added to the 3' end. DNA is bidirectional, meaning that strands are going to run in the opposite directions. That's also part of how the anti-parallel concept works. So if you have one strand that's like 5 to 3, the other one is going to be 3 to 5. And so quick question, RNA polymerase reads the template strand in the blank direction, but polymerizes the new DNA strand in the blank direction. B, we got B in the chat so far. What do we think? Yeah, exactly. That is right. Yeah. So the correct answer is that RNA polymerase reads the template strand in the 3 to 5' direction, but polymerizes the new strand in the 5' to 3' direction. This is a very, very important distinction. I cannot stress that enough. Super important that you know this.
Yeah.
Yeah. So
The phosphate.
yeah, basically because of the way the phosphodiester bond works, it can't be added to the 5' end because there's nothing for it to be added to. All you have is like the central piece with the ribose sugar. And you can't add, see how there's no OH groups there on the 5' end. There is nowhere for the incoming nucleotide to attach to. The only place it can go is the 3' end. That's where its bonding location is.
Do you see that up here? Yeah, so like it needs to have the hydroxide group to be added to the hexagon. So the 5' end, there's nothing for it to be added.
Okay, so from DNA to pre-mRNA to mature mRNA, this is the process of transcription through translation. So first, RNA polymerase makes the transcript of the gene that is being coded for in the DNA. This becomes a pre-mRNA. And so then before it is exported to the cytoplasm where it can interact with the ribosome and become translated to become a protein, it must undergo post-transcription modifications. Thank you. So this includes introns being spliced out. So keep in mind, introns are spliced out, exons stay in, introns are spliced out by the spliceosome. The pre-mRNA also has a 5' methylguanosine cap that gets added to it, and a 3' poly A tail that must be added. These are important for stability and for protection, also for nuclear export. If not, these mRNAs can be degraded before they make it out of the nucleus and to the ribosome. And so then once it has these things, it becomes a mature mRNA, and it is then exported from the nucleus to the cytoplasm where it can interact with the ribosome and eukaryotes. In prokaryotes, it's a bit differently because transcription and translation occur at the same time, and there's no nucleus, right? Yeah.
If one of those aren't added to the mRNA,
Mhm.
it can leave the nucleus.
It-
Or is it both of them?
It can leave the nucleus. It just won't make it.
Oh,
Is
okay.
the thing. So it needs the 5' guanine cap and it needs the 3' poly A tail for stability and for nuclear export. If it doesn't have these things, it's probably not going to make it. It'll just get degraded by the cell. No problem. Any other questions?
Do the 5' cap and the 3' poly A tail do anything else but stability and protection?
Really?
I would, yeah, that's all I'd be focused on for purposes of this.
And to
Yes.
Keep it from
Oh,
being
sorry.
eaten by, I forgot the name of the exact enzyme, but there are enzymes in the cell that eat RNA, and those
Um,
keep it from being eaten by them. I can't think of the name right now, so that,
RNases? Is it like what?
I don't know.
I forget.
But yeah,
what it
there are
is.
enzymes that will degrade RNA that's just free floating, so that keeps
Yeah.
it from being eaten by them.
Yes.
There's some function of the
Yes, so the UTRs, better known as the untranslated regions. So an important distinction to make, UTRs are transcribed. They are not translated, as their name suggests. So yeah, in the UTR is where UTR contains, I believe it contains the ribosome binding site. They do serve functions. Then you have like the polyadenylation signal sequence in the 3' UTR. But yes, the UTRs are involved a lot. I forget which type of transcription it is, if it's eukaryotic or prokaryotic, but things bind in the UTRs that make transcription stop, I believe, if I'm thinking of the right thing. So yes, they are useful.
They're also stabilizing just in general.
Right.
We don't really go into the detail on it, but
Okay, so RNA splicing, I kind of talked about this already. Introns, these are non-coding sequences. They got to get spliced out before the pre-mRNA can become mature mRNA and go to the ribosome. Exons stay in. The introns are spliced out by the spliceosome. This is a molecular machinery that is used to carry out splicing. That's really all you need to know for this. And then SNRNPs, which are small nuclear ribonuclear protein. This protein just base pairs with the intron at the start sequence of the intron and then cleaves it right there. This is also known as a ribozyme, an enzyme whose catalytic component is RNA. Any questions here?
Yeah. What's the SNRA RT?
It's a ribonuclear protein. I don't think so. No, just know that it can base pair with introns and just cut it out right there.
I don't.
I don't know. I don't know.
I would just know about as much as the exam question that had SMN.
Yeah.
Who's there?
Topic 4, translation. Okay, my last topic. Everyone's doing great. This is awesome. Okay, translation. So we just talked about transcription. Now we're going to talk about translation. So it is defined as the process by which mRNA transcripts, better known as mature mRNA transcripts, because they've been added with the 5' cap, the 3' poly A tail, are used as a template to create an amino acid chain, which can then become a protein. And that's the goal of all of this, to make proteins. So just remember that strings of amino acids are called polypeptides, and proteins are comprised of one or more of those polypeptides. All right, so what's involved in translation? You might guess the ribosome. It is responsible for protein synthesis. It makes the proteins within a cell. You have a large subunit and a small subunit that comes together. The small subunit binds first, just for the record, and then the large subunit comes in. These are made up of rRNA, which is ribosomal RNA, and proteins. And the RNA in the large subunit catalyzes the peptide bond formation. So once the small subunit is able to bind, the large subunit can come in and catalyze peptide bond formation. You have what's known as transfer RNAs or tRNAs. These bring the amino acids to the ribosome to be strung together. Each anticodon corresponds to the amino acid that it carries. So amino acids are going to have a codon. What the tRNAs do is they carry the anticodon, so the complementary sequence that can then be used to synthesize, or that will then pair to an amino acid that can then be used to create the primary structure of a protein, which is just the string of amino acids. There is a special type of synthetase called amino acyl tRNA synthetase. This is what covalently links the tRNA with the amino acid. The charged tRNA has an amino acid attached, uncharged does not.
Yeah, could you just go over really quick, there's a question on the exam, like ribosomes versus ribozyme.
Yeah, okay. So ribosomes here are what's responsible for like protein synthesis. They are a piece of cellular machinery. They're made up of rRNA and proteins. I think ribosomes were on a couple slides before, right?
Yeah.
Oh no, it's all good. So ribozyme, like these are enzymes whose catalytic component is RNA. So they cut the RNA to splice out introns. They are a form of cellular machinery that just cuts introns out.
Also known as the spliceosome.
Yeah.
No, it's okay. Any other questions here?
Can you explain what a charged tRNA is?
Like,
Yeah, so yeah, I can explain that. 1 second.
do you want-
No, it's okay. So charged amino acyl tRNA synthetases are enzymes that are going to add amino acids to the end of essentially the chain that is going through the ribosome. So that's what adds the new amino acids. Each tRNA matches a specific amino acid with a codon. So that's how like the codon versus anticodon complementarity works, or complementarity, excuse me. So that's basically the main role of the amino acyl tRNA synthetases. The charged ones bring in more amino acids. Yeah.
The anticodon that the tRNA carries doesn't code for the amino acid, right? It's the codon of the mRNA.
Yeah, that sounds right.
Yeah,
Okay.
so that's where using the codon table to code for amino
Yeah.
acids. So it's a good way to remember, like using the codon, the anticodon is just what pairs with the mRNA. So that's how it finds it.
Right.
Yeah, but it's like the mRNA itself that's coding for the codon.
Okay.
Yeah.
Okay, so mRNA is read in groups of 3 nucleotides. These are better known as codons. There are 64 codons that code for 20 amino acids. AUG is the start codon and it codes for methionine. AUG is like the start codon. It also sets the frame for codon reading. So you don't start reading. The reading frame does not begin until you read AUG. And then there are 3 stop codons: UAA, UAG, and UGA. These all signal stopping of translation, which causes the 2 ribosome pieces, the large subunit and the small subunit, to dissociate from each other. And oh, sorry.
No, I was just gonna answer the question in the chat.
Frequently, when ribosomes are not translating, they are dissociated from each other. So the large subunit will be somewhere and the small subunit will be somewhere. And then when they have to translate, they come together. But otherwise, they just dissociate.
Same with those tRNAs. To answer the question in the chat, I believe that once the amino acid comes off during the process of translation, it's just going to go back to the cytosol until it matches up with its same specific 3-letter sequence. So those tRNAs are sequence specific. So it's just going to be in the cytosol until it finds another sequence to bind to that matches what it has. It'll be uncharged.
Okay, so breaking down the process of translation, it can be broken down into initiation, elongation, and termination. So initiation is different for prokaryotes and eukaryotes. So in prokaryotes, the ribosome binding site, or the ribosome binds to the ribosome binding site, which the small subunit of the ribosome, I should say, binds to the ribosome binding site on the mRNA. There is an initiator tRNA carrying FMET, which is formal methionine. This is why the start codon is always AUG, because the initiator tRNA is always carrying formal methionine, which binds to the start codon. And so once that happened, the ribosome large subunit can bind, and then translation starts once FMET enters the P site. In eukaryotes, it's a little different. The initiator carrying the tRNA, carrying the formal methionine, binds the small subunit. The small subunit binds mRNA at the 5' guanine cap. The initiator tRNA scans for the start codon, and then once it's got it, the large subunit binds to that, and then translation begins. Next step is elongation. So the new tRNA whose anticodon matches the codon and the mRNA comes into the A site. It is worth noting that the formal methionine is the only amino acid that does not enter the ribosome through the A site. It enters through the P site. All incoming anticodons will enter through the A site, but are known as the active site. A peptide bond is formed between the formal methionine and the next amino acid in the chain that's in the A site. And then the ribosome is translocated along the mRNA, which is just a fancy way of saying that it moves, so that the amino acid in the P site moves to the E site, the amino acid in the A site moves to the P site, and a new amino acid brought in by tRNA can then enter the A site which will then form a peptide bond with the amino acid in the P site, and so on and so forth. Okay, last step is termination. So elongation occurs until the stop codon is reached and enters the A site. When that happens, release factors enter the A site. These catalyze the hydrolysis of bond formation between tRNA and the P site and the polypeptide chain, which is a fancy way of saying it tells the ribosome to stop and just, end translation and dissociate. The polypeptide then leaves, the empty tRNAs leave, and then the ribosome subunits dissociate and are ready to attach to another mRNA strand.
Are like are the amino acids that are like in the P and E site, do they finish being translated?
Yes, I believe so. Yeah, as long as it's not in the active site, it should be fine. Like as you can see in the middle diagram there, they get taken out. So I believe they get taken with it. Just because after the active site, that's really where the peptide bond formation occurs. So it's attached. So once stop codon comes in, doesn't matter what's in front of the stop codon, it's still attached to the polypeptide. Okay, almost done. Point mutations. These are really important. You got to know these. They range from being, not really affecting anything to being like catastrophic. Silent mutations, these don't change the amino acid sequence. The genotype changes, but the phenotype does not. What this is a single nucleotide change in the mRNA that will still code for the same amino acid. So there's literally no change. They're often undetected. That's why they're called silent. Next up is missense mutation. This changes the amino acid sequence. So this is where a single nucleotide change occurs and it changes the amino acid sequence. So if you have a cysteine and then a nucleotide change happens and you have a valine or literally anything else, that's changed amino acid structure, amino acid sequence. This will change your primary structure. This could have a lot of effect or it could have almost none. If you go from a very nonpolar amino acid to another very nonpolar amino acid, you're probably not going to see much change. But if you go from a polar amino acid to a very nonpolar amino acid, you are going to see a lot of change. It might cause your protein to not function and fold correctly. Because as we all know, structure determines function. Next up is nonsense mutation. This results in an early stop codon, so premature termination. The polypeptide is what's called truncated. It just means it's very small and probably can't function. What happens is a stop codon there's a mutation that changes the nucleotide so that it codes for a stop codon. And then probably the most significant one is the frame shift mutation. This is where an addition or deletion within the translation frame shifts the reading frame. So as mentioned, the reading frame is 3 nucleotides long. Each codon, it is determined by the start codon. So you have AUG, and then you have your next 3 nucleotides after AUG. That's your first codon after the stop codon. In a frame shift mutation, if you add or delete a nucleotide, the entire frame gets disrupted. This can cause stop codons to come early. This can cause stop codons to be missed completely. So you can either have a protein that's too small or a protein that's far too large.
Any questions on this? Because this is pretty important.
Yeah.
Probably gonna see some.
What if addition or deletion of a nucleotide causes an early stop codon? Would it still be a nonsense? So if in a frame shift mutation, so a frame shift mutation is categorized by an addition or deletion that shifts the reading frame. So if a early stop codon occurs, it depends on what causes it. If there is a nucleotide change, it's a nonsense mutation. If there is a nucleotide addition or deletion, it's a frame shift mutation. The actual location of the stop codon is less important. What matters is why it's there. Thank you.
So like, what's the difference between, can you just go over silent again? Okay.
Absolutely. Yeah. So as we all know, the idea in that silent mutation is that the nucleotide sequences form codons, and these codons correspond to anticodons that correspond to amino acids. That's why we have our codon table. So the silent mutation makes a single nucleotide change. Okay, within one codon. However, as we know, there are multiple codons that code for the same amino acid. So what happens is the nucleotide change occurs, but the amino acid does not change because the nucleotide that was there that was changed out is replaced with one that will still code for the same amino acid, just via a different codon. Make sense?
Yeah, thank you.
And if
Awesome.
we just look at these diagrams over here, you can see that the A on the top is changing to a T. And then if you compare that to the frame shift, the TA are being completely deleted. So that's like the main difference.
Okay, and I think my very last slide is for sickle cell anemia. This is a very famous case. It can be caused by a missense mutation in hemoglobin subunit where a glutamic acid is replaced by a valine. Glutamic acid is acidic. It is negatively charged and very polar because charges can be indicative of polarity. Valine is extremely hydrophobic and nonpolar. So as you can probably imagine, if you replace a nonpolar amino acid with a polar one, you have significant changes in your protein structure, which then causes significant change in protein function. The valine on surface in mutated hemoglobins reduces oxygen carrying capacity, it blocks blood flow, and it can poke holes in the cell, which causes cell lysis, and it's not good. And that is all for me.
Okay, so topic 5 is pretty hefty section. This is gonna be transcriptional regulation and the lac operon. Do we have any questions on the lac operon before we start, or anything from the last exam? I know that was like a pretty tricky question. No? Okay, let's get started. So transcription factors, TFs. There are 2 types of DNA binding proteins, and these would be sequence specific and non-sequence specific binding proteins. And a question that you might see would be like, why does RNA polymerase need to be a non-sequence specific DNA binding protein? Does anyone have a guess for this? Yeah. Yeah, so if it's going to read the DNA, it needs to be able to move down the DNA and not be stuck on one specific sequence. So yeah. So general transcription factors. In prokaryotes, these are called sigma factors, as we see in our lac operon example. One sigma binds one RNA polymerase and creates the RNA polymerase holoenzyme, which you can see up in the top corner. And this complex binds a promoter, but different bacteria have different numbers of sigma factors. E. coli, for example, the lac operon has 7 different sigma factors. And then for eukaryotes, they have basal transcription factors for their general non-sequence specific factors. These are complexes of proteins that assist RNA polymerase with getting to the promoter. So basal transcription factors first bind to the promoter, and then RNA polymerase is recruited and binds. This is an important detail that's like often forgotten for eukaryotes compared to prokaryotes, because we always think about the lac operon, but that's not a eukaryote. So these regulatory transcription factors help out.
So are general transcription factors non-sequence specific?
Yeah, they don't have a specific sequence. Okay, regulatory transcription factors. These are transcription factors that can have positive or negative control in transcription. So positive would help initiate transcription and negative would suppress transcription. So in this example, the activator helps transcription move along and the repressor leads to no transcription. Okay, sigma factors. These are sequence-specific DNA binding proteins that bind to 2 sequences in their promoters, which would be the -35 box and the -10 box. These are the consensus sequences that we talked about in class a bit. There was like an example problem that was kind of guessing what the consensus sequence would be. And to do that, you would just look at the most common letter at each spot in the genome that you're looking at. And then that most common letter is going to be what makes up the consensus sequence, like the tab box, TA. Okay, what is an operon? An operon is a set of genes on a DNA strand that can be transcribed into one mRNA. So we see this in the lac operon, and transcription is controlled by one promoter here. While the genes are transcribed separately, they have one promoter. It's important to note. This is really common in bacteria, prokaryotes. Oops, sorry. Yeah, this image doesn't really give you anything. So the lac operon components. So this is a you can have the LAC-I repressor, which is a protein coded for by the LAC operon. This is a transcriptional repressor. It codes for the LAC repressor protein, and it's produced at low constant levels of glucose. And then we have the promoter, which is LAC-P, and this is where sigma 70 binds once bound to RNA polymerase. So again, sigma 70 is going to bind to RNA polymerase, and then that holoenzyme is going to come down because this is in a prokaryote. And then the operator is LAC-O, and this is where the LAC repressor protein binds. And this is where it's bound if there's no lactose present in the cell. If there is lactose present in the cell, you're going to see the repressor bound to allolactose and the cytosol, which is like the same as saying lactose, and it's not going to be blocking transcription. And then the CAP site is where CAP and CAMP would bind, and this is the positive regulator of the operon, which we'll get more into. So there was a question on the exam that was asking about what proteins were transcribed in the DNA region given, and that would have been LAC-I, LAC-Z, LAC-Y, and LAC-A. So LAC-I was not transcribed by the LAC operon, but it was in that DNA sequence. Now that's why it was one of the proteins on that exam question. So near the operator, these three genes code for three separate proteins. LAC-Z codes for beta-galactosidase or beta-gal, and then LAC-Y codes for permease, and LAC-A codes for beta-gal transactylase, which you don't really need to know about. And then LAC-Z codes for beta-gal, and as I just said, and this helps split lactose into that glucose and galactose molecules, and it's needed for glycolysis to make ATP. And then permease is that channel protein that helps things get in and out. And then we don't really know what beta-gal really does. You don't need to know that. So a scenario where lactose is not present in the cell. This is when you're going to have the repressor bound to the operator because there's no lactose in the cell to take it off the operator. So it's going to be repressing. You don't need to produce these proteins to deal with lactose, and there's no lactose in the cell. So this would block RNA polymerase from transcribing the LAC operon genes, and the LAC operator is in the promoter region, as I just said. So why is the repressor bound to the operator when lactose is not present? Because again, you don't need to break down lactose when it's not in the cell. When lactose is present. So this is when lactose enters the cell, binds the repressor and takes it off the operator. The repressor changes shape, and that's why it leaves the operator. It no longer has affinity for the operator because the structure equals function. And then the removal of this repressor by lactose allows for RNA polymerase to bind to the sigma 70 and to begin DNA transcription. But lactose is not the only regulator of the LAC operon. We also have glucose, CAMP, and CAP. So sigma 70 itself has pretty low affinity for the promoter, and it needs something else to help it bind to that promoter. And we have CAP to do this. So when glucose is present with lactose, the cell does not prioritize LAC operon expression. And then, is there something in the channel? Okay.
Mhm.
So CAP is a sequence-specific DNA binding protein, and it helps stabilize RNA polymerase at the promoter. CAP is regulated by CAMP, which is a nucleotide, and CAP is needed to bind first to CAMP in order to bind the promoter and stabilize RNA polymerase. So CAMP binds to CAP, and then CAP binds to its DNA binding site, which is kind of upstream as the promoter, as we saw in that diagram on the exam, but it's in the promoter region, and then it helps stabilize RNA polymerase. So when glucose levels are high, CAMP levels are low, and then when glucose levels are low, CAMP levels are high because they have an inverse relationship. And a quick question would be like, is CAP a positive or negative regulator of the LAC operon? Yeah.
The sigma 70, that is
Yes.
Yeah.
so it's the same thing in eukaryotes or no?
No, like the LAC operon example is just a specific operon that's found in E. coli to digest lactose. So this doesn't apply to eukaryotes right now.
Also, prokaryotes themselves have operons because it's their way of clumping genes together where you have one promoter for multiple genes, whereas in eukaryotes, it's one promoter, one gene, one promoter, one gene.
Question? Oh, okay. Positive, yeah. So a little diagram of CAP and CAMP. In the absence of CAMP, CAP doesn't bind to the promoter. It needs CAMP in order to bind. And you can just see that happening down here, and that would be the cap and camp binding site. A question on the exam was talking about where would cap bind, what would it be bound to? It would be bound to camp and then its DNA binding site in the promoter region. And then all of this would be the lac operon. Okay, leaky expression. We don't cover this too much, but it is just important to note that things are not always bound where they're supposed to be bound. They're going to dissociate, they're going to move around, and the repressor can dissociate from DNA even without any lactose being bound to it. Just for a short period of time, it would just kind of jiggle away and come back. And this allows RNA polymerase to access the DNA and to keep those proteins that we need at low levels, because if you don't have those proteins in the first place, you're not going to be able to use the lac operon. And this occurs because you always need a little bit of beta-galin permease, like I just said. So there's never really zero expression unless you would have like a super repressor, something that we've seen on problem sets. So it would be kind of like a 1 level of transcription at all times. Okay, so getting into some more different transcription factors, we have the basal transcription factors. This is the general transcription factor used in eukaryotic cells. As we briefly mentioned, we're going to get more into it. So this basal transcription complex is made up of a number of proteins, and they bind at the TATA, the TATA binding protein spot. And this is just like an AT region. And as we talked about, well, Joe talked about, these are easier to break apart as well, but to unwind the DNA. So if RNA polymerase comes down at a region high with A's and T's, it's going to be easier to rip apart. So that's kind of why you have that consensus sequence. And then these basal transcription factors bind to the promoter first and then recruit RNA polymerase. This is different from the holoenzyme we saw in something like prokaryotic cells like the lac operon. So these would bind to the promoter, the DNA first, and then RNA polymerase comes in. Okay, enhancers and silencers. These are terms used to describe the areas of the DNA where there's positive and negative transcription factors. An enhancer is a place in the DNA where positive regulatory factors bind, and this would help stabilize RNA polymerase binding to the promoter, and it would help cause transcription to occur. Again, it's an enhancer, so it's going to upregulate. And one would bind upstream of the promoter, and then one would bind downstream of the entire gene. And you don't really need to know that, but there's some images on like problem sets of that. And then silencers are places in the DNA where negative regulators bind, and these block RNA polymerase from binding to the promoter because they're going to want to decrease the amount of transcription. They're a silencer. Okay, any questions on this? So DNA bending and combination control. So these are, it's kind of a weird word, but these are needed because transcription factors bind far away from a promoter in these eukaryotic cells. So DNA bending allows the distal control elements, which just means they're far away, to come in contact with RNA polymerase in order to actually have an effect on RNA polymerase. So DNA bending proteins are sequence specific. As we can see, that little purple thing would have to be sequence specific in order to bend it at the right area to get those distal elements in contact with RNA polymerase. Eukaryotes use different combinations of transcription factors to control the transcription of many genes, because we would have to have so many different transcription factors if they were specific for each gene, because we're way more complex organisms than eukaryotic cell. And that would just reduce the amount needed.
Is that what we eukaryotes?
What?
Like the DNA bending with the distal control?
Yeah, these are all eukaryotic things we're talking about right now. Yeah. For the bending protein? Okay, yeah. So we just have so many different genes in our cells that if we had a transcription factor for each one, it would just be too many. So what we do is we bend at different portions in the DNA to get different distal control elements in contact with RNA polymerase. Okay, so here's a little practice question. If DNA bending proteins contain a mutation that lowers its affinity for DNA itself surrounding gene X, how would this affect levels of transcription in gene X? And think about the different types of control and how each type of enhancer or silencer could affect this. Yeah.
Bringing like an enhanced research to the archives, then less of the should be expressed.
Mhm.
But it was silence with the house.
Yeah, exactly. Yeah. So this is kind of a tricky question because some of us would immediately think enhancers and some of us would immediately think silencers, but we really want to consider both. And it's exactly what she just said. If the DNA bending protein would bind to the DNA surrounding the gene less often, this bring the distal control elements less often. So if the distal control element was an enhancer and the enhancer is coming in contact with RNA polymerase less often, you're going to have less transcription. But if this was a silencer and it's coming into contact with RNA polymerase less often, you would have more transcription. That makes sense. Okay. Oh, I didn't realize that was it.
Yeah.
Okay.
All right, so that's what we have. Thank you so much for being here. So what we can do is we have a little bit of time to stick around and answer questions. We can answer questions. If you don't have any questions, feel free to, yeah, bye. And otherwise, the recording will continue. Should I continue?
Depends on the question.
Yeah, actually, let's end it. If people are going to ask like individual questions, we can end it. So yeah, thanks for being here.